welcome to our viewers and listeners to another episode of Reflections on the History of Modeling and Theory. I'll remind everyone that this is part of some of the programs run by the Society for Modeling and Theory in Population Biology. That's the SMTPB, relatively new society, and our goals include providing a forum for discussing theoretical issues and results, strengthening the connection of practitioners to journals in our field, building this community through meetings, workshops, and online events. I think this counts as an online event of a certain kind. And maybe most importantly, we want to foster the careers of modelers and theoreticians in population biology. So this podcast is part of those activities as one in a series of informal conversations with leading figures who have helped to shape our field, and also who can at this point reflect upon the changes in modeling and biology over the course of their careers. So with the technical wizardry of Kaylee Charlebois, a graduate student at UBC, Sally, Otto, and I are delighted to welcome Joan Roughgarden. Joan obtained her Bachelors of Arts in Philosophy in 1968 at the University of Rochester, and then her PhD in Biology 3 years later from Harvard University.
And it's interesting that she brings that combination of both philosophical and biological expertise. You can see that in a lot of her works and her books. Her thesis was actually on the implications of density-dependent natural selection. And James, I think it really paved the approach of integrating competition and ecological processes with natural selection.
Mm-hmm.
I first came to understand her approach through her 1979 textbook, Theory of Population Genetics and Evolutionary Ecology, an Introduction. And it's a classic. It's a wonderful book to learn about how to think synthetically about eco-evo processes and how to model them.
So just to go through a bit of Joan's biography since graduating, she began at the University of Massachusetts in Boston and then moved to Stanford for really the majority of her career. Sally's kind of alluded to some of the impact of Joan's work and how it combines different flavors of theory, and it also combines theory with empirical work. And that includes decades of work on anolis lizard biogeography, behavior and competition in the Caribbean. And then more recently, Joan's turned to understanding the life history of microbes and viruses, exploring, among other things, how selection and collective inheritance shape microbiome-host integration.
Yeah, and that's a recent paper in AmNat, which is quite interesting about how does evolution work and how do we understand integration? Is it competition or natural selection shaping it?
And thinking about some of the awards throughout Joan's career, recognizing much of that work, she's a fellow of the American Academy of Arts and Sciences, has also been a Guggenheim Foundation fellow, and also has won an award for her, I guess writing is something we do all the time as scientists, but I guess for writing maybe a little bit outside of the kind of journal literature. She won the Stonewall Prize for nonfiction from the American Library Association, and that was for Evolution's Rainbow: Diversity, Gender, and Sexuality in Nature and People.
And so now we welcome Joan. It's such a pleasure to see you.
Well, thank you. It's wonderful to be here.
Hi, Joan.
We're going to launch into our first questions. We've shared these with you. And the first question is really, when did you realize that quantitative methods had an important role to play in biology?
Well, I was taking my first ecology course, as I was a bio major at the University of Rochester. And the instructor was Conrad Istok. And and it was quite clear from the content of the course that there already was a presence for quantitative methods. And what was interesting is how much difficulty he had, and I mean this in a nice way, explaining it. And then I knew how this all could work, and so then I could explain it. And I thought that I might have a potential as someone who could explain math to biologists. And so I thought I'd be able to make a living doing that.
I learned so much from your 1979 book. Thank you. It's really
Hmm.
where I got a strong sense of modeling from.
Oh, good. Yeah. So that's where it was.
Uh,
And I didn't start it, of course. When I came into the field, there already was a quantitative presence here. The
right.
only other field that did was neurobiology. And so I sort of had, if you wanted to do a part of biology, which was quantitative, you really had two choices, it seemed to me. One was ecology and evolution, the other was neurobiology. And if you did neurobiology, then you're going to be in the lab all your life. And so if you did ecology, you could be outdoors,
Mhm.
swimming with sharks or whatever.
So I don't know if it was the outdoors or if there were specific people who inspired you to take the ecology and evolution direction rather than neurobiology, but were there people who did inspire you early on in your career?
Yeah. Well, there was Conrad Istok who taught the ecology course, and I didn't take that knowing particularly what it was. It was just part of the curriculum. But the very first course as a freshman I took, and it was taught by microbiologist named Wolf Vishniak, and who has long since passed away. And he talked about microbes in a very sensitive way. He made like microbes come alive, the microbes that live in deep sea vents and in Antarctica and things like that. And that gave me an appreciation for microbes as organisms in a sense. But what was particularly helpful was that I came from a family where no one had ever gone to university. And so I was at the University of Rochester on a scholarship. And and there were other people at the University of Rochester were kids who came from New York City, often from schools, from prep schools and so forth, and they were extremely confident, and I was
Yep.
very unconfident. And and so when we did the lab, and he had Professor Vishniak asked a very tricky question on the lab final, and I remember seeing that and so getting the answer correct, whereas all these hotshots just glossed over it.
Um,
And then on the day that the grades came out, they were posted on the wall. And then I looked up my student number and there was an A in the course and I couldn't believe it. So I went away, had a cup of coffee, came back to check whether or not it was still did I misread this? And some of these hot shots were getting A minuses and B pluses and things like that. And that's when I said, you might be able to do this.
well, and you come back to thinking a lot about microbes in your recent work.
As it happens, yes. Uh, yes. I think I may have mentioned that it was sort of by invitation. I was giving a lecture in Israel at a conference, and I was lecturing on bird social behavior and parent-offspring conflict and cooperation and that sort of stuff. And so afterwards, this guy comes up to me, and he had the manner of a New Yorker. As it turns out, he was raised in New York, and
Hmm.
was an Israeli, but he rolls up to me and says, "What do you have against microbes?" And I said to him, "Nothing. I never think about them." And so he then said, "Well, we really need some theory for microbial processes and so on." And they were into this idea called the whole genome, which is the union of the genes in the microbes with the genes in the host. And they had sort of some verbal conjectures about this, and they were getting beaten up in the literature because they had really no grounds for these assertions, and they just sort of almost wanting things to
Yeah.
happen. And so then I started working on that problem, and I was just going to be a technician. I was just going to put equations toward their ideas. I was going to embed their ideas in some equations for them. And so my first paper in this area was with them as co-authors. Um, But then, I started to get more involved. And so that's been about 6 years now or so that I've been working on
Yeah.
microbes and viruses now.
Well, you can
Joan,
see,
just to sorry, Sally, go ahead.
oh, just that how important that eco-evo, you cannot separate those when you're thinking about microbial interactions. Yeah.
No.
Right. Joan, that's kind of I guess almost full circle from Professor Vishniak to your current work. I'm
Yeah.
wondering if you could tell us a little more about maybe as you started to do research in ecology and evolution, were there other mentors or people whose work inspired you at that stage as you were kind of building your career?
Well, Robert MacArthur, of course, was a really key figure. I met him shortly before he died. He was jaundiced at that time and had yellow complexion and so forth. And I do remember. He was at Princeton, and I had just published my first paper on density-dependent selection, which provided a population genetic basis to the ideas of R selection and K selection that he had
Right.
ginned up with Richard Levins, if I recall correctly. And so he was very happy about that. And then when I looked at him, what I could see, even though his health was failing, is his eyes glistened. He had a brilliance to his eyes. And the only person I've seen, or at least seen pictures of like that, is the cellist, Stavros. I forget how to pronounce his first name, but Richter, something like Stanislaw Richter, or he's a pianist, I think. And but he also has those eyes that just stare out at you, and so. Um, he was an inspiring man, and so he brought conceptualization to ecology. And you could say, of course, Hutchinson did earlier, and so did David Lack. So I mean, you have to give credit where credit's due, even before that. But he did it, of course, mathematically, and uh, so he was a really big uh, big influence on me. And I remember when I was a grad student at Harvard, and so my first advisor there was um, George Clark, I think was his name, or something like that. He was like an old-time ecologist, and
Mhm.
uh, Larry Slobodkin used to define ecology as the misidentification of beetles on a field trip. That's sort of what it was. You go to a stream, you catch some bugs, and then you misidentify them. And that's sort of all there was to ecology. And George, I mean, they were nice folks, but then I wound up switching to a different advisor. And Robert MacArthur was really key to the field, and of course his and also Richard Levins's view was of ecology together with evolution. So they thought of evolutionary ecology at that time.
And was that where the evolution influence or direction started?
No, well, I suppose in a way, but for me personally, I got really involved in the integration when I was still a student and working through the derivations of the basic equations. So I remember working on the derivation of the model for one locus and two alleles in population genetics, and realizing that the survivorship coefficient L, could be density dependent. I mean,
Right.
after
Right.
all, genes are not disembodied things in the air. They're in organisms, and the organisms are growing as a population. So right off the bat, yeah, I must have been either a senior or first year grad student or something like that. And that's then what led to the first paper of density dependent selection, because of just doing the derivation. And there it was, right in front of me, that they were together. They
Mm.
were in, inseparable, almost synonymous. I mean,
Mm.
um, and then it turns it, then, you know, um, historically, evolution and ecology, have been separate and have separate societies and so forth. So that has been, for the entirety of my career, that's been a hindrance. And so the American naturalist, the American Society of Naturalists, was from the beginning the only one that would sort of do both at the same time, but evolution and ecology wouldn't. And I remember when Paul Ehrlich and I wrote a textbook on principles of ecology, and we had sections in there on evolution as well as sort of typical ecological topics. I remember giving a lecture at might have been Washington State or something like that, up in the Northwest, and asking the question came up about textbooks, and he said they didn't want to adopt the textbook Paul and I wrote because it had both evolution and ecology in it, and they wanted a different textbook for the evolution course and a different textbook for the ecology course, and so ours wouldn't really fit, and so the pressure to keep them separate, has been extraordinary over the years.
Right. And mistaken.
Yeah, and mistaken too. Yes.
One of the questions that I would like to pose is whether there was an aha moment in your career where you were muddled or just unsure about something and you did a model and then all of a sudden you were like, aha, I understand this now.
Well, there have been lots of them, but I think that the one that I kind of liked, the most aha moment was
The biggest aha.
The biggest aha was when I was doing modeling of barnacle recruitment in the Rocky Intertidal Zone. And so that was the first time I started making brand new models as opposed to adapting models from the Lac Volterra tradition and so on. So the models, the marine models, were brand new from scratch. And so I really liked the idea of how that model predicted sort of the dynamics of the settlement onto rocky surfaces. Then what we did was, so we thought we understood the process pretty well. So in doing this ecology, you can take advantage of remote sensing to get information on where the water is going, because the basic story is that the larvae go out into the ocean, they accumulate on little zones between upwelled water and water that has come from the north. So off the California coast, water is coming from Alaska south, or actually from Oregon south. That's offshore water and it's hotter. But then when the winds blow, you get upwelled water adjacent to the coast, which is saltier and colder. And where they come together, you get a front. And the larvae that are released from the rocks are carried out to the front and hang out at the front. So you can actually, on a boat, go over the front and with a net, actually catch larvae right at the front. And so when the winds drop, this whole front moves back to shore. And then when it collides with the shore, it dumps a whole lot of larvae on the shore, giving you a so-called recruitment pulse. And so now you can predict when this is going to occur by using the data from buoys. So there are offshore buoys which are measuring water, measuring currents and wind speed and all sorts of stuff. And also you can get satellite data because there's a sea surface temperature signature of these fronts, so you can see them there. So what we did was to take our basic model for the settlement on the rocks and then assimilate data on a daily basis from the buoys. And so they're assimilated into the coefficients of the model. And so in that way, make a quantitatively accurate model to predict recruitment. And it was kind of neat having a really well-calibrated model in nature, because usually we don't do that, and that's too expensive to do that, and so on. And in fact, once we published it, some people said, well, we already understand this process. This is overkill.
Ha.
But it was interesting to see that you could make an ecological model completely quantitative and accurate.
Wow.
So anyway,
That
Where
is
did
it.
that come out?
I'd have to check my CV. I don't remember exactly
Yeah.
where. Um,
Because you've actually had a lot of papers on barnacles and
yeah,
recruitment over the
yeah,
years. Yeah.
yeah,
It's funny you say that about the people saying it was overkill, but there are a lot of ecological models which never get really well parameterized with data
Yeah.
at all.
yes.
And so pushing things in the other direction, that seems like a really good aha moment, and seeing
Yeah.
how it works.
Yeah, you can do it. It can be done.
Right.
I mean,
Right.
it's not clear that that's the best use of a model, because I think models, mostly useful for telling stories, and then you proceed to see whether it's a true story. But
But
uh,
maybe every once in a while, having
yeah,
checking your understanding of the system quantitatively, empirically, is actually
yeah,
useful, not just in that particular study, but in other projects you're doing.
yeah, and also it wasn't done in the lab, so that we would parameterize this out in the field.
Right.
Yeah,
Amazing.
So, as I said, your work in the area is broadly very well known, and you're allowed to pick a paper in this area for this next question if you want to as well. That's also fine. But we're wondering if there's a favorite paper of yours that you wish was better known, and and what is it you like about that paper?
yeah. So I actually checked my CV and found it. It's in 2009. I wrote a paper called "Is There a General Theory in Ecology?"
Mm.
That's published in the journal Biology and Philosophy. And I wish that were better known, because there's a lot of misunderstanding as to whether ecology can have a general theory, what shape it would be if we did have one. Do we want one? And what do we give up if we have a general theory in terms of our authenticity in each place? And
Mm-hmm.
and so I think that article goes into sort of the pros and cons of this. I mean, it's a question. We could choose to have a general theory, and we could frame everything as departures from a central theory. Um, and sometimes, population genetics and evolutionary theory, tends to look as though it's a unified theory with branches and so on. And we could frame ecology to do that. If we wanted to. So the question is whether we want to. And I think that paper goes into the pros and cons of whether uh, theory in ecology is a general theory.
Ooh, I want to I have to read that. I haven't read that before. Do you want to give us a spoiler? What do you feel? Because I that's an interesting perspective. Is you can
Are
either
you looking for a yes/no answer, Sally, to
yeah,
that, or?
well, I find it interesting that what you're saying is we could either have a null hypothesis or a null kind of theory from which we break it or we branch it out. But maybe that constrains us. Maybe
Yeah.
we then try to shove too much into that core theory.
No, well, I mean, the way ecology's tended to unfold is that we have an atlas of models, you might
Yeah.
say.
Yeah,
So
yeah.
and we have the notion of biomes, and so and uh, and and then you can also deconstruct whether or not evolutionary biology is as general as represented sometimes, because
Exactly.
it
Right.
puts a lot under the rug.
Mhm.
Uh, and so no, I don't have personally, I don't have much of a view on it. I'm sort of take it as it comes.
All right. We'll have to read the paper.
Yeah,
That's right. And Joan, I
Um,
think that was the paper I had maybe seen you give at a conference around
that's
then. I
right. I
remember
gave that.
that, and it stuck with me. I was at a Gordon research conference, I think.
What a coincidence.
Yeah, that's right.
Um, Joan, many, many, many years ago, I was one of your students in your ecology, theoretical ecology class, I think. But I want to fast forward us to now and ask, what advice do you have for students who are interested in learning at the interface of theory and empirical data?
Um, well, I do have an opinion here. I'm not sure, how popular it will be, but I think that theoreticians actually have to do the field work, and that they can't rely on collaboration. They can bring collaborators in to help, but the best examples I can have is that when I was doing the field work, I was taking data that a field worker normally wouldn't take,
Right,
because
right.
it's only if you know the model, you know what parameters you need to get data on. And
Mhm.
if you don't have a model, then you just get whatever has been traditionally got. And
Right.
Right.
and I remember for the stuff I was doing on competition with lizards, uh, anybody, in looking at competition theory knows you have the competition coefficients, and so you can measure those, which you normally have measured in terms of the difference in body size between the animals. But then the other parameters involve the carrying capacities.
Mhm.
And so that means you have to take data on the carrying capacities. Well, normally people don't do that. They just look at the birds and the difference in the sizes of the birds, or the lizards, the difference of the sizes. But you have to take data on the amount of insects there, and then also especially for lizards, which are of course not warm-blooded, you need to know the distribution of microhabitats within the environment. So we came up with this idea of using little cylinders, which we call gray body objects. So that would integrate, so we would put them around the habitat. And
Mhm.
then we would go with a thermocouple and measure the temperature in each one of those. And those little cylinders would integrate the convection, the radiation, and all that to indicate what the temperature would be of something sitting there. And
Hmm.
so then you could get a distribution of thermal microhabitats in the environment. And so that, together with the uh, insect data, told you what the carrying capacity would be um, in the habitat. And as I say, normally biologists wouldn't take those data at
Right.
all. And similarly, when we were doing the barnacle work, I already mentioned the buoys. We used buoys and we used the satellite data, but we also introduced a new technology, which was radars. Radar, you can set up radars along the coast, and they send out a signal that bounces off the waves, and you get a Doppler shift. And from that, you can measure, and then the Doppler shift includes the speed with which the waves itself are moving, as well as the speed with which the underlying water that the waves are on are moving. And so then you subtract one from the other, and you can actually get the currents out of it. So we could
Mm.
then get
Mm.
a picture of the currents, uh, which were then leading to the recruitment and so on. And again, all the rocky intertidal people just do the same experiment over and over again. They build a cage, they exclude this animal or that animal or that animal, and we
Right.
don't get anywhere. And so it's only, I think, when theoreticians get into the field and take responsibility to make sure that the data that are needed are gotten. And they may not be the
Mhm.
best skilled at doing this, because field people usually are better at technique in the field and so forth. But without
Mm.
that kind of very intimate involvement, I don't think the connection will be made.
Some of that is
Fantastic advice.
advice, Joan, but what is your sense of the way students are trained, or are students being trained to do both, or to be
Mm.
theorists but also in the field? Is that
No,
happening? Is it happening? Okay.
no, no, they're not, of course. And since I was a student, of course, the mathematics has gotten more sophisticated in certain areas, and so it is a specialization. But and you want to say you can't do both. I mean, but you have to do both.
Mhm.
Hmm.
Yeah, I think that's very well put. It's and I've witnessed it a bit that you just approach things in a different way. You ask things in a different way, and you have to collect things in a different way. Well, if you're going to want the data, you have to then
Yeah.
get it yourself.
Um, this next question, you've already, I think, to some extent answered. But I'll ask the question, maybe give you a chance to riff on it in a slightly different way. We've talked already a bit about ecological theory and evolutionary theory being separate fields almost historically, and then becoming more intertwined. And I guess the part of the answer you've already given is they were always intertwined from day one. Um, so I guess maybe the way I could ask this question would be, what do you think does give rise to that resistance for integrating ecology and evolution? You mentioned it being a very strong kind of resistance early on, and maybe still there to an extent now. Why is that?
Um, well, I think it has to do with a suspicion of the type of the degree of rigor that can be realized in the field. So ecologists are usually identified with doing field work, and of course, geneticists have pioneered a lot of techniques with laboratory work with Drosophila, going back to Dobzhansky and so forth, and so people who work in the lab as opposed to the field tend to think that field work is uncontrolled and not rigorous. And so, therefore, you really can't find out what's
Mm.
going on in the field. They're skeptical that you can actually determine what's happening outside. Um, it's not only, I mean, this is very general. I remember when I was just starting at my lab at the Marine Station down at Stanford's Marine Station off of California, there was an invertebrate zoologist down there, Don Abbott, and he was never very sympathetic to ecology. He didn't want to look at organisms on rocks and so forth. His inclination was always to collect the animal and look at it in the lab. And it's that uh, issue of being
Mhm.
suspicious of the tractability of the
Mhm.
outdoors versus the tractability of the lab, of the indoors. And
Yeah,
of course, the ecologists always say, well, yeah, you may have nailed it in the lab, but what does that have to do with nature? So that's what
right.
their comeback always was. And uh, so I just think that with time, as the capability of field work improves, the suspicion starts to drop a little bit. In fact, I remember for my tenure seminar of all things, I had just finished doing experimental work with Steve Picella at the time uh, about competition between lizard species. So we had these cages outside, and we put different lizards in and so forth, and detected the competition between anolis lizards. And Pat Jones, who was an immunobiologist and one of the molecular biology faculty, said, "Oh my God, they do experiments."
Ha.
And so she was really impressed, and sort of helped in my tenure process that we actually did do experiments outdoors. And of course, now experiments are pretty commonplace. But I think with the increased skill of field work, um, the suspicion is going to drop a bit. And meanwhile, but the appeal of laboratory work isn't going to go away. And especially for microorganisms and so on, that's where you have to do it. Um, and whoops. So uh, I think so. I think that's my answer. So I think
Yeah.
it's that suspicion.
See,
Yeah, I was going to say that last. Sorry, Sally, go ahead.
there's another element in there, and that has to do with control. And I think one of the
Right.
things that is appealing about theory is you're fully in control.
Yeah.
But then what do you learn? What do you lose learning about if you are not in control and the environment is wreaking havoc with your best laid plans? Do you find that a little bit that when you're in the field, you have to let go a little bit of controlling everything?
Well, yeah, you're right. And you don't even think about control very much. You set up the experiment and you let it run, and whatever
Yeah.
happens, happens. But and I remember there were different definitions of what it meant to do an experiment outside. The When I was doing the oceanography work and had I was working with biological oceanographers, they call, quote, an experiment uh, the deploying of instruments at a certain location.
Oh,
And
interesting.
yeah, and so they're not going to control the
Right.
Gulf Stream or anything like that. The whole
Right.
issue of control just never comes up. But on the other hand, then they can control where they set up station and watch it.
One last thought on that connection, Joan. That I thought you were going to go to back to microbes at the end there, because I would say that the lab experiments have become much more ecological for microbial communities.
Yeah,
But
yeah.
I still heard the complaint that they're not realistic enough, or they're not similar enough to natural communities, particularly in terms of maybe the abiotic variables or variation. But that's maybe a place where those areas are converging a bit more than in the past, is for microbial communities in the lab and the field.
Yeah, maybe. And though my feeling at the moment about the lab experiments for microbial population dynamics and so on is that they tend to look at equilibrium conditions. And
Mm-hmm.
so they solve for steady states and
Yeah.
they try to experimentally replicate those. But I think in nature, microbial populations are usually uh, either in a bust or a boom or something like that. And I think that I'm very suspicious about how much density dependence there actually is. And I think and therefore, in the modeling I'm doing, I tend to be modeling bacterial populations in the log phase while they're still growing rather than when they hit
Mm.
the stationary phase.
Mm.
Um, and but that will be sorted out in time. That's where I'm coming from on that, is that if they're always in bust boom, then you should really be talking about an exponentially growing or an exponentially declining population rather than a steady state population.
And island biogeography and movement and my colonization as well, I would imagine.
Yeah,
Not,
yeah.
yeah.
Well,
Interesting.
we're at our last question, Sally. Sad. It's always sad to get to the end of an
Yeah.
interview, but here
Yeah.
we are.
Joan, what do you see as the major open questions in theoretical ecology or theoretical biology more generally?
So yeah, I jotted down a couple. I mean, and of course, I can't read my notes. So the so the first thing is this introduction of ecological thinking into microbiology.
Mhm.
And this may be a little self-serving because this is what I'm currently working in, but I think that we need to found or form an offshoot or an extension of ecology that would be called theoretical microbiology. And it
Mm.
seems to me this subject area is really wide open, and we can't just take existing community models and so forth and population dynamic models and plug them in because the phenomena are so different. And there's no real counterpart of the lytic to lysogenic transition in a non-virus. And
Mhm.
there's really no real counterpart of um, the contrast between vertical versus horizontal um, uh, my colonization. So that if you're looking at the population dynamics, for example, of a microbiome within a host, there could be vertical transmission of the microbiome or horizontal transmission of the microbiome. And a consideration like that doesn't really come up if you're talking about population dynamics of birds invertebrates or plants and so on. And then there's what the notion of a holobiont is, which would be I take a neighborhood idea here, like similar to Wright's neighborhood idea in genetics, that it's the space around an animal that the animal controls. And so it includes the stuff inside it, but it also might include the immediate domain around it, which is particularly relevant for plants and the stuff in the root matrix. And then there's the coevolution of these viruses and microbes, and then you have tripartite issues where you have a virus and a microbe and a host, and there's just so many issues here, and it's possible to do foundational work in theoretical microbiology. And so I think that's one of the big areas where we can do a lot. Then two other areas which are closely related is I think the field still is defensive about sexual selection and about the problems with it. And there are a whole lot of problems with it. And you might take the position that it could be fixed up, but it seems to me to be impossible to fix it up because it starts out on the wrong track with Darwin's original focus on mating success and so on. And obviously, natural selection depends on the offspring delivery, not on mating per se. And of course, a sexual selection person will obviously say, well, we know that it depends ultimately on getting offspring into the next generation. But let's talk about mating success anyway. And so it keeps coming back to that. And so the whole supposition that there's a hierarchy of genetic quality, and that even if there were, could a female detect the differences and so on. And I think that the field still needs to confront these issues and not to lash out with personal complaints, with personal attacks when these problems are brought up. So the field will not progress, in my view, until it non-defensively starts addressing these issues. And as you may know, I have been championing some alternative theories for these, but for these issues. But the real issue is getting an open critical introspection in the sexual selection area. And then similar or related to that is the notion of the selfish gene, and whether or not that has outlived its usefulness by a long shot. And I think it has, and I think we have to move past that because it privileges competition. Now, when you look at behavior of animals and so forth, yes, they could be competing, but yes, they could be cooperating a priori. They're the two possibilities. And by cooperation, I don't mean altruism. I mean they're working together. And so there'd be a lot of behavior possible, a lot of behaviors needed to make cooperation work. They have to stay on the same page. They have to be coordinated. They have to perceive a common goal, so forth. And so there would be behaviors that could bring this about, a lot of which involves physical contact and the physical exchange of pleasure. And so I think that the whole selfish gene premise is not neutral. It's not theoretically neutral.
Right.
And we need to replace that with a theoretically neutral starting point. And I think that's the other main question. So they're the three things I think are important. Sort of launching theoretical microbiology, and then having a critical introspection in sexual selection, and then replacing the selfish gene with a theoretically neutral counterpart.
Words matter, and even the phrasing, yes. And you were focusing on how there aren't isolated genes, there are interactions between individuals, but there are also interactions between the genes, and there is no a gene does not exist on its own.
Yeah.
It's not transmitted on its own. So focusing on it as if it were isolated
Yeah. Well,
misses the interactions.
I think young theoreticians now have a lot to look forward to. And also, your email to me a while ago asked about new tools as well. And
Yes.
I do think that AI can be helpful, primarily to speed up coding.
Mm,
When I think about how long it takes to do theory, it used to take quite a while to actually, on pencil and paper, do the analysis, and it was mistake prone as well. And now, so when Mathematica came out, that speeded up stuff a whole lot. But
Right.
Mathematica still has a very difficult syntax, and I think some AI might
yeah.
help with producing code fragments and so on much faster. But the rate limiting step is still thinking, the time it takes to think. And because like right now, I can work on a model and then just type a little bit and I get an answer. And I said, geez, now let me think about this.
Right.
And
Well, that's what makes being a theoretician fun, right? It wouldn't be
yeah,
fun without that rate limiting step.
that's right. That'll stay the rate limiting step because you can't speed that up.
You can't ask AI to figure it out for you. Yeah.
No.
Not yet.
Joan, thank you so much. Is there anything else that you wanted to touch upon or share with students in the field?
Uh, well, no, just things look pretty good. But the other, yeah, in terms of looking into ourselves, but the other thing too is that we're of course all worried about the national and international situation and the attacks on universities, and I think it's important to remember, and if it gets really bad, okay, and the universities collapse, the NSF collapses and everything, and we're all on the street selling uh, selling food, or
Mhm.
the way the way they do in Madagascar or someplace. You can just walk along and everyone's just kind of making making it do. But it reminds me of the French Revolution, and not I mean the French Resistance,
Yeah.
uh, where you'd have cells of people who would know things, and it's ultimately we are the repository of knowledge.
Mhm.
We no one can take away what we know from us, and we can stay together, and we could even have dinners with one another if it comes to that, in which it's okay to talk about science and it's okay to talk about an equation, and we can keep knowledge alive even if the shit really hits the fan. And I think that's something we have to be prepared to do.
Mm. There's power in that, in holding on to our values and our thoughts
Yeah. And
and what's
our
most,
knowledge, they
yeah,
can't take that away from us.
right.
And to value how it's transmitted as well, so that it
Yeah.
isn't
Yeah.
lost.
That we share it.
Yeah.
Well, thank you so much for sharing your knowledge with us today, Joan.
Thanks,
Well, that's
Joan.
right. And good luck with with the new society. It sounds great.
Thank
Okay.
you.
Take care. All right. Bye-bye.
Thanks a lot. Bye.
Bye.